Hi everyone, I am Nadia Bihari Padhan from IISc Bangalore, and I am working with Professor Rahul Pandit. And I'm going to talk about activity-induced self-propulsion and self-deformation of a droplet. And more specifically, it is motility-induced phase separation in an elastic confinement. And this is our ongoing project. And last month we started this. So your comments and criticisms are will valuable for us. So what is motility-induced phase separations? Simply, phase separation in active matter system is MIPS. Okay. So for example, here in this case, okay, so there's a phase separation of muscle beds. So it was uniformly distributed at t equal to 0, and it separated into different clusters. And another example from a simulation from Michael Cates group from active model H, and this is phase separation for contractile swimmers, and at a particular length scale they arrest the coarsening. So there is hydrodynamic interactions between the swimmers. So what we are going to do is we ask the questions: what happens to the MIPS if it is confined in an elastic boundary, and that boundary can deform also and it can propel also, like it's flexible. So before going to technical details, I want to show one of our simulations, then I will go in detail. So what we're going to do is, let's take a droplet of one fluid, and the droplet is in another fluid, and what we are going to do is we are putting active swimmers inside the droplet and see how they are phase separating it, and what is the effect in this confined boundary, like your droplet boundary is confined boundary. So phi is my representation of droplet binary fluid mixtures, and psi is some active scalar field which represents some, let's say 1 is the dense region and -1 is the dilute region, and omega is the vorticity field here. So if you start from a homogeneous distribution of motile swimmers inside the droplet, then they are doing phase separation, they are doing self-organization, and finally the boundary or your droplet is propelling in a particular direction. This is from one of our simulations. So I'll talk more details right now about this. So we write down the models, like we start from two order parameters, color order parameters. One is phi, which captures the droplet interface, and another is psi, which captures the active matter. So starting from a free energy functional, so this is your double well potential, like in model H or cantilevered system, and this is for phi field, and similarly we write down for the psi field here, and there is a coupling term between phi and psi, and that beta coupling coefficient which we take greater than 0, so which ensures there is attractive interactions between phi and psi. And like in model H, these two terms are interfacial energy term. So they try to form, they like interface, and these two terms they don't like interface, and there is a competition, and they form interfaces of finite thickness, epsilon1 and epsilon2, and sigma1, sigma2 are their respective sufficiency coefficients.
These phi and psi, they are both related to density field, or
yeah. So yeah, so phi. I'm going to explain here. So phi is a smooth field, it's not. So it represents, if it is +1, then it's fluid 1, and if it is -1, then it is fluid 2, and there is a diffuse interface between them with thickness this one. And for the psi also, you can think of like this. For +1, it's high dense region, and -1 is dilute region of the bacterial suspension. So we write down the equations, conserved equations. These are conserved order parameters. The first one is for the phi field. This is sort of Cahn-Hilliard equation only, and this ensures del squared term or nabla squared term ensures the conservation. And this is for this case, this is chemical potential, like is the functional derivative of with respect to phi. And finally, this is the form, and similarly, it follows for the psi field also. So we talk about one coupling, beta. Now, when we write down the solvent equation or Navier-Stokes equations, so there are two stress coming. One is passive stress. We are calling it passive because in terms of active matter terminology, that it is from the droplet boundary, and this is coming from the gradient in bacterial density. So I want to point out something here. So for this phi case, sigma 1 here, that is diffusive sufficiency, and sigma 1 here, that is mechanical sufficiency, that is the reaction to the fluid. So they are equal here, but in the active case, or for the active field psi, sigma 2 is not equal to sigma 2 tilde. So this is the difference. And sigma 2 tilde greater than 0 signifies, like, captures action size swimmers, they are pushers, and sigma 2 tilde less than 0 for contractile swimmers. And all these literatures are available in this paper, Active Model H. Okay, so we are going to solve all these three nonlinear equations using some numerical methods. We start from a box size 2 pi across 2 pi, and we do direct numerical simulation. We don't do any approximation to the equations. We directly solve those equations. We use pseudo-spectral method to solve these equations, and for time integration, we use some semi-implicit exponential time differences on RK2 method. And we do high performance computing using CUDA-C, and our all simulations are carried out in Ampere 100 and Volta 100 GPU machines. Those are recent GPU clusters, and our simulations are about 400 times faster than their serial CPU counterpart with some optimizations. And for this problem, MIPS inside a droplet, we take this initial condition phi. So it's a circle, and with +1 inside and -1 outside. And for psi, it's outside it is -1, but inside the droplet we start from a random perturbation to 0, that is homogeneous state, +1, 0.1 is the magnitude of that perturbation. And initially we take vorticity field to be 0, and we define some dimensionless numbers here. One is Reynolds number, which is inertia divided by your viscous term. So you notice some RMS velocity of the droplet, and R0 is the initial radius of the droplet, and Peclet number, which is the ratio of the advection to the phi field or psi field divided by your diffusion mobility and sufficiency. And we define something called activity number, which is the ratio between your sigma 2 tilde. This is, and we are interested in negative sigma tilde 2. This is for contractile swimmers. So what happens when there is no coupling in Navier-Stokes equation? The activity is 0 in this case. So it's normal binary fluid mixture phase separation. It will go to a complete phase separation at the end. And if you increase the activity number is 0.15 and Rans number is very low here, it's almost 0. And Peclet number is 3. So the movie I showed in the beginning, it's self-organized and it propels in a particular direction. And if you increase the activity number, then it's kind of random. They are moving in a random direction with random velocities. So what is happening is this is all coarsening arrest. Like we are not allowing phase separation to happen. So at a particular point, we are arresting the phase separation. So we define some length scale like this, and for the normal low activity 0 case, it's either 1/4. It's unlike 1/3 in normal periodic box. This is well-known Lipschitz-Ryalov exponent 1/3, but here inside the droplet it is not getting enough space for the phase separation, so it is slower than that 1/4. And if you increase the activity, then the length scale, so you can see it get arrested at a particular length scale and time scale. Yeah. So if you see the plot for the center of mass velocity of the droplet, for the low activity case, it's kind of periodic, and for high activity it looks random. And if you see the deformation, it's a measure of the perimeter. It's also random for like very high activity. So the questions we are trying to, or we will ask, like it's an ongoing project. So what is the mechanism of self-propulsion here? Like how to explain that? We are thinking of to calculate pressure, to looking at the pressure distribution inside the droplet to explain that. And what is the route to chaos? You are increasing, then it leading to a chaotic system. So these are the two major questions we are going to ask in future. And I want to summarize. We are able to study the MIPS in a droplet by using two scalar order parameters. And the coarsening due to diffusion gets arrested by active stress. And our DNS shows the self-propulsion and self-deformation of the droplet due to MIPS. Thank you very much for the attention.
A couple of things. One, nice talk, nice work. Why did you want two scalar fields to have this whole thing? Why you could just have one scalar field? See, after all, phase separation is you've got a background fluid medium and you have some other composition variable in it, some solute, some particle, whatever it is. It condenses, and if it condenses, but it has an incompatibility between its interfacial tension in the chemical potential and interfacial tension in the stress, you should already see the phenomena you're talking about. So I didn't really understand why you had two fields.
Can we capture the interface, like droplet interface kind of thing, with single order parameter, like the bacterial density or active matter plus the interface of the droplet?
One field is to produce the droplet,
yeah.
The other is to produce MIPS inside. That's why you're doing it. I see. Okay. Other thing, you were asking about the mechanism for the motility.
Yes.
Have you looked at the flow fields inside the drop?
So we looked at the
typical sperma flow fields.
No, actually, we looked at the pressure distribution.
Yeah, just why didn't you look at the flow field inside the drop?
Yeah, but yeah.
No, I mean, typically when a drop swims, actually what happens is you get this squirmer field with a surface flow going back and then something going inside.
Inside the droplet, you get it?
Yeah,
Okay.
inside the droplet all the way to the surface. I'm almost sure if your droplet is swimming that way, you will find that kind of a field.
Okay.
And that will be, I mean, I'm especially given that you have these little blips going forward and all this stuff, I think it'll be there.
Yeah, so yeah, we'll look at that. Thank you for asking questions.
Can I ask? Yeah, very interesting. But your this growth picture that you have plotted, your data are very noisy. And
No,
the
at,
difference between 1/3 and 1/4 is very small, actually. So probably. Yeah,
so it's I mean to say that it is lower than 1/3. It's not 1/3. So I fitted 1/4. So it's a small droplet, so the noise is coming, like its domain size is less.
Okay, so I think we should go to the next talk. Thanks.
Thank you. Thank you.